All right, to wrap up our talk on cellular respiration, I want you guys taking a look at this diagram. It is absolutely critical for helping us understand it. As long as we can work through it, there's a lot to this diagram. So help me out. Let's first identify the three major steps: glycolysis, Krebs cycle, and the electron transport chain. Now let's take them one at a time. Glycolysis, what was our reactant? What did we put in? Glucose. And at the end of it, if you look at our outcome box, we had three different things that we made. Pyruvate, 2 of them. We made some NADH by doing redox reactions, and we got 2 of them out of glycolysis. We also ended up making 2 ATP. So here's what we put in, glucose, and those are the 3 things we got out. Now, ladies and gentlemen, the 2 ATP, let's start keeping track of how many ATP we're making total in this process. So 2 net for glycolysis. Now, even though it doesn't, don't forget about the transition step. That transition step to get into the mitochondria gave us a couple things. It gave us 2 more NADH. It released a couple CO2 molecules. And it made our 2 acetyl-CoAs that let us get into the Krebs cycle. So remember, those acetyl-CoAs, that's what we turned the pyruvate into, and that's what got us into the mitochondria. The CO2, we just give that off as waste. But the NADH, remember, we were stockpiling that. We take the NADH from glycolysis along with the NADH made here, and we're sending them on to that last step. We wanted to make quite a bit more of that, though, and that's where we're at with the Krebs cycle. So for the Krebs cycle, we put in our acetyl-CoA. What did we get out of it? 6 NADH and 2 FADH. And those continued on to, again, that 3rd step. We also made a couple more ATP in that process. So we're going to add that up here. Now, ladies and gentlemen, that brought us to the 3rd step, the electron transport chain. Remember, the whole point of step 1 and step 2 was to, make a little ATP, but mainly this NADH, this FADH. We wanted all that stockpile to drop off here. And what we were able to do with that by running through the electron transport chain was make most of our ATP. We made about 32 approximately. So that brings the molecule to be about 36. Now, just a couple things to keep in mind. Where does all of this fit in to the overall respiration formula? We don't want to lose sight of that. We said way back when that if you gave a mitochondria glucose and oxygen, that we would be making CO2, H2O, and ATP. Don't forget our balanced equation. You put 6's before everything except glucose and ATP. So we want to make sure that we kept track of all this. Where is all of that stuff? Well, guys, in green, I'm going to go ahead and circle our reactants. Where were those? Glucose, right here. We've got that one taken care of. But what about the oxygen? Where was that? Oxygen didn't come in until the very, very, very last step. Remember, it was our final electron acceptor. So one thing we needed for the ETC was that oxygen. So that's one thing that was put in. Final electron acceptor. Remember, the whole process stops if we're not able to have some oxygen there. So let's go to the other side now. Our CO2 molecules. I'm going to switch over to black. we've got 2 CO2 molecules here, but what about the rest? If you go back and look at our output boxes, we also gave off 4 CO2 during the Krebs cycle. That takes care of our 6 carbon dioxides. Now, ladies and gentlemen, the water, when we had oxygen accepting those electrons, it also took on some protons. And so water is a byproduct of the ETC. And that's where that came in. ATP made in all 3 steps. So we don't want to lose sight of our overall formula. If you can give a cell the glucose at the very beginning and some oxygen at the very end, it will run through this process, creating ATP, our main goal. We also end up giving off a little bit of water and CO2 as byproducts. Not stuff we really need. Our goal was the energy, the ATP.